So first, thank you for having me. My name is Brian Walsh. I think that description of what I do is probably a little bit polished for what I actually do. I came to this world in a very different manner. I don't have training in entomology. I don't have a lot of formal training at all. Um, Only ever got an associate's degree, but for several decades, I ran a landscape contracting business that I owned. And I was lucky enough to be just a few miles from where Spotted Lanternfly landed in the United States. And as a commercial pesticide licensed business in Pennsylvania, we took notice pretty quickly. This was new, this was different. Um, and I got involved and wound up working for Penn State doing this. And I think a better way of wrapping up a lot of how Fred described me would be cat wrangler. I do have some ability to, as a contractor and having had employees for many years, I can wrangle the cats and organize them. And I love the researchers that I work with. Researchers aren't always the most the absent-minded professor comes to mind sometimes as we're trying to get things to get off the ground. But um, this is just a quick update. I tried to pull out some of the more what I think are interesting things of what's going on in lanternfly world. It's really come a long way and really not far enough in the last 10 years. So if you have questions, throw them up in the chat and or at the end I'll take them. Like I said, I was minding my own business. I had my landscape contracting business and we were going along. And then in 2014, late in 2014, in November, this hit the news that lanternfly was found in Pike and District townships, and literally on the line between the two townships. And we started paying attention. And at a public meeting I went to in 2015, uh, this is the takeaway. It can't really fly. It was more of a hop and glide. So don't park near trees so it doesn't land in your vehicle and you don't accidentally move it, but it couldn't fly well at all. So it was pretty much limited in how far it could spread. The eggs were laid within 6 feet of the ground, also under objects, rocks, natural, man-made, it didn't matter. And that it must take a meal from Melanthus altissima, otherwise known as the tree of heaven, tree from hell is a more apt description of it, which in and of itself is an invasive in the United States. And it had to take that meal to complete its development from nymph to adult. And the other thing was that nymphs and adults walk up and down trees daily, and so you could use these sticky bands wrapped around the trees in order to uh, get to stick them, literally stick them and trap them and kill them on the tree trunks. And some of this information had come from South Korea, where it was an invasive early in, I think it was about 2003, 2004 also. And so some of the things that we were hearing was from the South Korean literature. And by 2016, in my backyard, and that was a distance of 7 miles. And that was a little bit of an eyebrow raise because for something that can't fly, there were some serious numbers that made it there. And there was still some insistence that they were hitchhiking. They must have been hitchhiking with careless people. By 2017, it hit in mass. This is a silver maple tree in my backyard, injected with dinotephuron. And you see the open red. You generally don't see those open wings unless they're flying or they're startled, or if they ingest dinotephuron, it will make them lose control and the wings will open. And on the ground, you can see beneath the tree by August, it was pretty clean. By September, it was a pretty good pile. By October, that pile just kept growing. And this is because the dinotephuron, the insects kept coming in. They feed, they ingest the dinotephuron, they die. And that's how the systemic insecticide works. And it was pretty apparent that these numbers were not small, that were feeding on this tree and being killed, or you can see they're still twitching from the dinotephuron, and that pile got pretty deep. In not this tree, but another tree that we injected and threw a tarp out on the ground, we killed 13,000 off of one silver maple in a week's time. And
Holy
so
smoke.
those numbers are significant. The population has expanded very quickly. That's the side of my barn there, and all those black dots you see are flying lanternflies, and they're actually really difficult to film. You have to have just the right cloud background to catch them. But you can see that's not a hop and glide. And so, so now the alarm bells started going off, and what was being said, unfortunately, in the early days has persisted as a myth, and it continued on. We were taking down a neighbor's tree, and at 70 feet off the ground, we were finding egg masses. So that 6 foot within the ground was obviously not true. I think nobody bothered to go up and check higher was my feeling. But the Atlantis requirement, by now I had been working with some of the Penn State researchers. I'd been on the county board for Montgomery County Extension and uh, working with some of the Penn State entomology researchers, we decided to set up this experiment where we had these cages with trees planted in the ground. And the reason we had to plant trees in the ground is that spotted lanternflies are just incredibly difficult to keep alive in a colony. They are voracious feeders. It can be done. The methodology is getting better. In the beginning, it was atrocious. They feed so heavily that you have to constantly rotate in nursery stock for them to feed on, or they just will not survive and or survive just barely. And so we had the idea, well, let's put the trees in the ground, let them grow, and put the insects in these bigger cages where they can move about freely. And in those cages, half of those cages have the tree of heaven in it, and half don't have that. And the other species in there were ones that we commonly saw them feed on, which were maple, black walnut, and let's see, river birch and willow. And and lo and behold, after the first year, this chart on the left, we had 46 egg masses. First time we ever had egg masses laid in captivity. 46 with Atlantis and six without. And so certainly there were more laid with Atlantis with the tree of heaven present, but definitively there were eggs present without there being tree of heaven there. And this following year, the trees were more mature. They supported much stronger numbers, 96 to 14 egg masses at about seven times the difference. Um, uh, Between the with and without the tree of heaven. And the important takeaway is tree of heaven is not the lack of tree of heaven is not going to keep them from successfully reproducing. Those eggs went on to be very viable. They hatched out. No problem seen by not having access to that tree of heaven. And some folks have taken this experiment to mean that, well, obviously with the tree of heaven present, that they are more successful, that they are going to be much stronger population numbers. And I will caution with that to say that just because they do better with tree of heaven in this particular study, there are only four other species of tree there and not the entire range of diet that they have access to in the wild. And these are the true prolificous feeders. They will feed on anything except conifers. Conifers, they'll probe them, they don't like them, but pretty much anything else, and that's right down to grasses and weeds, especially when in their nymphs, but also as adults. If they're hungry, they'll feed on it. And so that question about the tree of heaven, it persists. I still see it published. I still see it published in national news that it has to have that tree of heaven. We know it doesn't have to have it. Does it promote fitness? Maybe, but we haven't systematically eliminated all the other possible combinations. We do also seem to find the new populations in new areas where that tree of heaven is present. And, because it advances the fitness or because they congregate there because they really like it as a preferred host. And to me, it's not much of a difference in the question. It's a chicken or the egg. We do find them there. Atlantis is very good for scouting. It is a preferred host, but it is absolutely 100% not necessary. And that myth just keeps persisting. So, going back to that early messaging from 2015, a lot of it was wrong. A couple things were okay. And what it more realistically looks like is that they can fly. They're not strong flyers, they're not agile flyers, but they can fly. Quite frequently, they will hop and glide. Quite frequently, they will walk rather than fly. but they can fly. The eggs are laid anywhere, especially under objects, being natural man-made. They like to be under a sheltered ledge, particularly under tree limbs. Look on underneath the lateral branches, and you'll that's where you'll tend to find them. Sometimes on the trunk, but mostly under a sheltered area. Atlantis altissima is a preferred host for the entire life range from nymph to adult, as is grapevine. And that becomes, as we'll get into that, and that's where we're at now. It's a very big problem for the grape industry, but it does remain a host from nymph to adult, where some other species are not preferred at different life stages. And we also know that nymphs walk up the trees. They rarely walk down the trees. They jump out, they disperse out. We rarely catch them from the top side of a sticky band, always from the bottom. And sticky bands are good for catching nymphs, not so good for adults because the adults can fly or hop right over them. So 10 years later, what do we know? Well, starting with the basics, it's Lycorma delicatula, it's Fulgoridae, and that is in the Hemiptera family. And in that Hemiptera family, um, it's there's going to be some interesting things that come along with that being hemipteran. So where is it at? Earlier on, we were talking about Indiana. It's more than a couple border counties now. This is the map that's maintained by Cornell IPM. They update it with some frequency. You can see Maryland pretty much completely populated. Pennsylvania, now we've just added a couple more counties. I think at this point we only have 8 left. But it's been marching its spread out from where it started in that southeast corner, north of Philadelphia. It's been moving out, and these big jumps, these broad jumps that are not contiguous with existing populations, those are almost certainly um, come about because of hitchhiking on human conveyances, and they're very good at hitchhiking. And in the life cycle, it's univoltine, 1 generation per year. It overwinters as the eggs, very nondescript, an egg mass about maybe the size of a quarter, a little bit narrower than a quarter, uh, 30 to 50 eggs in that egg mass. About 32 has been the running average. They will hatch out in the springtime. Right now, they're hatching out like crazy in Maryland, Pennsylvania. I would expect in the next week or two where I am. And the first through third instars are black with white spots, and about a quarter of an inch. They're about the size of a pencil point when they first hatch, and they roughly double in size every 2 weeks when they molt. The first three instars are black and white. The fourth instar gains that red coloring that's very distinct, and they're up to about a half an inch in size, maybe slightly more than that. And that's really when they start becoming noticeable. The only thing, their first three instars, sometimes people mistake them as ticks or spiders, but if you go to touch them, they spring. They are plant hoppers. And the fourth instars, the only thing that looks like that on the East Coast that I can think of would be box elder bugs, but box elder bugs have wings and lanternfly nymphs do not. And they will molt into adults and gain their wings. Generally in our area, it's end of July, that mid-July to end of July that happens. Maryland a little bit sooner. They hatch earlier. In Maryland, the further south you go, the earlier they hatch, and it's all by accumulated degree days. Univoltine, where there's a killing freeze. We have no idea really what's going to happen when it gets to areas where there is no killing freeze. It's actually now recently, that wasn't shown on that map, been positively identified in Fulton County, Georgia, which does not always get solid freeze, and certainly some areas in California, if it gets there, when it gets there, it'll be interesting to see what happens. But Univoltine for now on the East Coast in the Mid-Atlantic. It's hemipteran. It's got a fused proboscis. It feeds on the phloem of the plants underneath the bark, and it's got that fused proboscis that's tucked away when they're moving about, and it looks like an extra leg down between the front two legs when they are feeding. Has these nice orange antenna on both sides underneath the eyes, and those antennae can extend out when they're sensing things. And also, the quick identifier, male and female, you'll see in the picture on the right, that last segment of the abdomen, I believe called pectora, is bright red, dark red when it's a female, black when it's a male. Easiest way to sex them out. And sometimes after she's laid eggs, that'll be covered up with some of the waxy covering from the eggs. You have to wipe it off and check the color, but very easy way to tell what you have, male or female, with the adults. And as I mentioned, it's a great hitchhiker. This is magnified 65 times, and you can see that there's an aurelia pad and there's two tarsal claws at the end of each leg. Those aurelia pads, those suction cups, um, just give them the ability to hang on to, walk on glass like a housefly, hang on vehicles. This is why they're so good at hitchhiking. They can hang on the hood of a moving car. On my truck, 35 is about the speed that they've topped out at. And before they
Huh.
get blown off. But you can imagine that underneath the vehicle, if they're up in the frame rail, when they're not being buffeted by the wind, they can go the full duration and not be blown out of the vehicle from underneath. So because of that, they are very, very good at moving those big population jumps across states, across state lines on human conveyances, on the materials that we move. Also, the eggs are frequently laid on all kinds of things. Like I mentioned, even though they don't eat conifers, they will happily lay their eggs. You can see Christmas tree growers. This is a problem for them. Sticking your head inside a spruce tree is not the most comfortable thing to do, to look for these little tiny, what looks like blotches of mud. Tiniest twigs will lay on to the biggest trees. Sometimes the nursery stock, it's the container that they lay on, not the plant material. Tires, light bulbs, you name it. If it's a firm surface, they'll lay eggs on it. And we as humans move them around and give them a free ride to a new home. Like I said, the eggs are hatching in May for our area. A little bit sooner. You're in late April in Maryland. When they first hatch, they are this whitish color. You can see the two eye spots. They emerge from the holes in the egg cases, and they will just sit around and skeletarize. They'll harden up their exoskeleton for a little bit, and then they right up into the trees. They will move and they will start feeding wherever they're at and then continue to move. And they move when they're not feeding. This is a difficult bug to target with treatments. They are in motion when they're not feeding. Some amount of chill increases the hatch success rate, but it's not necessary. And I'll show you in a little bit later why cold is not going to be enough around here to kill eggs. Where you would have to be to get pretty much no hatch would be in Winnipeg in Manitoba. And we know this because Dr. Amanda Rowe and Anna Turblin from the Canadian Forest Service took egg masses from Berks County, Pennsylvania, exposed them to different periods of extreme cold and different durations of that extreme cold. And running them down in Celsius still trips me up, but the minus 15, I believe, is minus 5 Fahrenheit. And uh, after those different exposure times, it's either going to be extreme cold for a short period or continual cold. Basically, what happened was with a 49% hatch rate with no cold exposure, which is actually kind of on the low side, you can see that the only time that they didn't get the extreme cold was when they exceeded 15 degree or 15 days at the minus 4 Fahrenheit. We're never going to see that on the East Coast. They had successful hatch at 5 degrees Fahrenheit, minus 15 Celsius for 10 days. They still were getting hatch. We're never going to see that for extended period of cold on the East Coast up through the Northeast. And so we can just take that off the table that we're going to have a very cold winter and it's going to knock back the populations of spotted lanternflies even to get down to 5 degrees Fahrenheit for an extended period. And keep in mind, that is the entire thermal mass that those eggs are laid on has to be down to that temperature to see that. We're just not going to see it on the East Coast. We're not going to see it through most of the lower 48 states. The only place, like I said, would be Winnipeg that consistently sees that minus 27 Celsius more than 10 times per year to get to that kind of chill. And pretty much not much else in Canada is going to be exempt from egg masses being able to survive. So if they're going to do well in Canada or they're going to be able to survive in Canada, they're going to be able to survive in the lower 48. So puts that whole concept to rest. That was a myth that was kind of floated early on that a good cold snap would kill the eggs. It's not going to do it. So first through third instars, they like to feed on all kinds of tender materials. They tend to go for the new growth in plants, roses, both domestic and multiflora rose, that wonderful invasive are good indicators. If you have roses on a property and you're scouting for lanternflies, look for the roses. If you're going to have them in the first and second instars, they're likely going to be there. But you can see on hibiscus plants, red twig dogwood, red osier dogwood, they're happy as long as that material is tender and they can get their mouth parts into it. First instars will actually pierce through the leaf to feed. And as they move up in later instars, they tend to feed on through the stems. But if you can name it and it's not a conifer, they're going to be happy to feed on it, take a meal and go away. And one of the things that we've since learned from the early days is that they really don't like our fruit trees. There was great concern that our tree fruits were in danger, the peaches, the apples. They don't particularly care for them. A regular spray regime in an orchard will take care of them. Our orchard growers are not concerned at this point about them. It's a different story with grapes. They are deadly to grapes. Um, but our natural forests of native trees, not really impacted. They will move through, but they don't spend a lot of time there. And where they do tend to seem to be making their best living is in the areas of the intertie, the suburban landscapes, when they have unmanaged right of ways that generally have a lot of invasive plant species and underbrush. They like wild grapevines as well, but it's that mixture of those different areas where we seem to see them making their best living. And this particular site in Berks County is topped in. There was 50 red maples planted right up that main avenue and 50 red maples planted to the same variety at the same time, same size as wonderful replication for research. And I spent a lot of time out there monitoring the populations. And we do know that red maple, silver maple are very preferred hosts late in the adult season, just prior to egg laying and during egg laying. And what's really interesting is that, and I'll show you this, they don't stay there for the whole season when the eggs hatch. And so um, plenty of eggs were present. This is what it looked like. I just want to point out, you see these little white, looks like tags hanging out. Those are indication of fresh hatch out of the eggs. They will dry up within a week or two, but you can see that a lot of these other eggs don't have those tags. Those are the previous year's eggs, and so they will persist. The eggs will persist year over year for whatever reason. They do tend to lay eggs where previous eggs were laid or where another lanternfly has already laid eggs for the season. They tend to group them together, not always as a hard and fast rule, but generally there is some amount of aggregation, congregation with the egg laying that we see consistently. And but you can quickly tell the old eggs from the new eggs, particularly when they're fresh hatched, by that little white tag hanging out. And so where those eggs were in that site in Topton, we started monitoring, and we only looked at the first 8 feet up into the trees because the first instars are so tiny, and we could consistently see accurately the first 8 feet up. And our first trip out there in June 13th, 2020, across those 50 trees, we counted just over 1,400 uh, nymphs that were in the trees, in those 50 trees. By the time we came back for the 4th of July, because we were doing another large experiment with some Bavaria bassiana for big area-wide control. We came back in 4th of July, and there were some second instar nymphs there, but pretty much nothing else. The nymphs just were not in those maple trees, and look, this is what it looks like looking up the road in those maple trees. And if you notice off to the right, there's these leatherleaf viburnum bushes there. And sure enough, that's where the nymphs were feeding. They were feeding on the new tissue of the expanded growth of the viburnums all over the place. Uh, we found all, not that they were all there, but they were plenty of them in the viburnums and not in the maples where the adults had been feeding in the fall. And looking back at that map, you'll see this little access road out here in the field, and that's where we parked. So we were away from the trees, and I didn't want to carry hitchhikers with us, and went back to the truck, and the truck was covered with these lanternfly nymphs all over the top, the back. They encountered the truck, they climbed right up, just like it was a tree. And it really doesn't make a whole lot of sense when you look out here. This is all wheat. This was wheat field that year. The middle field, I believe, was later planted in corn. But the question was, why are these insects that have to have trees to feed on? Why would they be heading out into a wheat field? Well, they can happily feed on all the grasses and weeds that were out there, including the wheat. We went out and started looking in the field. They're not a big threat, but especially when you get a nice juicy weed growing up in the middle of that winter wheat, there they were. So they don't see those areas that are not trees and not shrubs as an impediment. They see them as a highway and may not be their favorite thing to eat, but they will take that meal and move on. And that gives them the ability to range very far and very wide. And that was something that was not understood in the beginning. There were a number of folks that believed that they had to have those only trees. They could only feed on trees. And that's absolutely not true. And so going back later, we continued counting, and you can see as late as August 8th, we counted a whole number of one. And then we started seeing the adults tick up in towards the end of August. They ticked up and they went down, and then there was this sudden spike at the end of September where our population just absolutely ballooned on that site. And again, this is only in the first 8 feet, physically counting each one, but we could also see much further. The adults are a little over an inch in length. We could see right up through those trees, and we did time 2-minute counts. And when you look at what was up above that 8 foot, those numbers got pretty big. We saw that jump from 509 on those 50 trees to the 8,100 and then 12,400 the following week across those 50 trees in that 2 week period. And what takes place is the uh, the fall equinox, and that's when egg laying starts. So we saw that population move in right at the equinox, roughly at the end of September, and that's where egg laying takes place. It tends to take place. And we also killed about 9,500 in that site with insecticide trials. So closer to about 22,000 that we saw influx in that 2 week period. That's why this bug gets so much attention. That's why this bug is such a freak out factor for the general public. I know entomologists tend to be enthralled by it, but for folks that go to work in the morning and then come home and see a tree crawling with inch-long insects, it can be a bit overwhelming. And so we see that the populations fluctuate at one location in one season within the season, and then from season over season, we see these population fluctuations. You see the dark green line was in the 2020 accounts, and the lighter green line there, that was the 2021 counts. The population went way down, and we're not entirely sure why that was. It followed the same pattern. They were there early as nymphs, and we had more time, more counts in the early part of the season than the previous year, but the adults ticked up a little bit later in 2021. And then in 2022, you see this, I don't know, call it a shamrock green line. Population went up pretty good. Not as good as it was, not as high as it was in 2020, but we did see a significant uptick over 2021. And so this is telling us that these populations are not going to stay continually the same. The best I can kind of approximate it to now is what we're seeing is Japanese beetles. Some years you see a lot, some years you don't, and that can change just in a short distance. And again, relatively close locations, in a nursery not too far away in Berks County, they were absolutely inundated. 2019, 2020, 2018 was really bad for them. And what we did was we injected, you can see the yellow stars here. Those were maple trees that we injected with dinotefuran and put tarps out to catch what fell out. And then we purposely put in uh, poles because uh, there's no pheromones that have been identified with this insect. I'm told by my colleagues that no Fulgoridae have any pheromones identified with them. Um, with with pheromones, there's been a couple pheromones that have been identified that work okay in the lab. They do not work in the wild, and we don't have an attractant for this bug. But we do know, and what we see the behavior over and over again is that they are attracted to vertical objects in the horizon. They will veer towards them when they're flying, and they will climb them, and they will climb right up those poles, get right up to the top, and launch themselves off again. They'll try and probe along the way for food, and it makes sense. It would make sense that they would be attracted to that because on that vertical object on the horizon might be a feeding possibility as a tree. Whatever reason they do it, we know that they're attracted to these vertical shapes. And so to take advantage of that, we purposely installed poles into this nursery and we wrapped them with delta methrin impregnated netting. And with the idea that they would go climb up the netting and get exposure to that netting and potentially be killed. And that's the netting that we've been using. We've tested a number of them, different manufacturers. This one is from Vestergaard, and this is anti-malarial bed netting used throughout third world countries and relatively safe for mammals, birds, and actually extremely safe for mammals and birds, uh, deadly to insects. And so we did some bioassays with it, and we found that with as little as 1 minute of exposure, within 96 hours, we were at 100% mortality, or 99.99%. And 5 minutes, we were moribund. Our insects were moribund within an hour. And that worked out fantastic, except for the fact that they were climbing up those poles and launching off and dying off in these topiaries and junipers, which then we'd have to like go out and try and find them. So we tried to come up with a way to keep them in place a little bit longer, and I did that with peony cages. Got my Wile E. Coyote going, and thankfully no rocket packs involved. But we just made it so that they could climb up, still hang on. They do not like to climb down. They would hang on the netting inside that cage around the outside, and then they would fall down dead. And it worked. These are all dead lanternflies on the ground on the tarp after the first day, and we did pretty good. With our 8 traps, we killed 12,773. Not as good as the 5 maples. They killed 15,000, but when you think about the amount of biomass and size of those maples, quite a bit more substantial than those 8 poles. But all told, the combined treatments, we killed just about 28,000 in that nursery without putting a ton of insecticide out into the environment. With that, from there, it developed out. We engineered this out. Randall Bach from Penn State Ag Engineering. We developed these traps that were non-conductive that could go on utility poles, and we worked with Pico Energy in Philadelphia. I absolutely did not put my children to work in the driveway building the mounting brackets because that wouldn't be right. And we have we had put them out around the city of Philadelphia. This is below the airport. This is in the docks, and wanted to see how would it work. And it worked well in some areas where it was the dominant height feature that they could see on the horizon. In areas like the ports, there was too much visual background. They all caught something, but these ones in the city didn't do so good. But going back to that nursery that was inundated, uh, you know, the numbers were pretty high there in 2019, 2020. By 2021, they'd dropped off substantially. and I'm missing a slide there to show you, but the number dropped significantly to the point where consistently 21 was down, 22, it was down so far it doesn't even show up on the graph. And when we took the traps down in 2023, we only got 150 out of this trap. So we went from that high of almost 13,000 to 150. Uh, but in 2021, where our numbers were down so far, just 3.5 miles away, that is honeydew. That is honeydew raining out of an ailanthus tree because the population was so high. Um, it's it's pocketed. The numbers jump around a lot. And another 6.5 miles in a different direction from that nursery in 2022, where in 2022 in the nursery, the numbers don't even make it onto the graph, the population numbers. We were comparing different nymph trapping techniques, and in there, and what the trial was this is a bug barrier brand on the right where the sticky part is facing inwards. In the center is just giant fly paper cut down with a screen to keep the birds off. And on the left is a modified circle trap. You can build that yourself with window screen. You can buy it. The fly trap many people use. Unfortunately, they don't always use the screening to keep the birds off. Bug barrier is a commercial product, is very effective. And what we wanted to know in this trial, that's 6 miles from the nursery that had almost nothing. We wanted to know how sensitive they were because different agencies around the country and internationally are using these traps to try and monitor to as an early warning system for lanternflies. And so the design of this trial was we had the different trap types at the bottom, and then we put a second trap, which was just the standard sticky band above it, to see how many would walk past the first trap and get caught in the second trap. What was the most sensitive trap? And you see this picture on the upper left. We had to keep that down to only a 24-hour exposure time because the bands would fill up and then they would just walk across the dead ones. But in 27 trees where the traps were out, on that site, by the end of the year, we trapped 80,000 insects.
Oh,
So 80,000 insects killed off of that site. And also, if you look, we were doing live counts, and because they're not very good at catching adults. But if you look at the live counts before the leaves fell off, once the leaves drop off, they move away. There's not enough sap flow, is the suspicion. We were still counting 1,300 live adults in those trees after killing 80,000 off of those trees. So as a practical way of controlling the population, bans aren't going to do it. And we had started all the way back in May. I mean, first instars, we took out 13,800. I mean, it's insane. And to still have those numbers at the end of the season. So, as a monitoring device, yeah, the traps can work pretty good. You can get a presence, positive or negative. As a feel good, it's good for homeowners to not kill themselves with insecticides. It's a good thing to do. You're not doing a lot of damage to the environment as long as you're not killing birds inadvertently and other wildlife. But which one worked the best? We found that the modified circle traps were the least effective. And what this graph represents is each set of vertical lines is 1 week of sampling. And if you start at 0% and to 100% on the left at the top, where that circle falls is is what percent were caught in the first trap that didn't make it to the second trap. And so you can see that the circle traps at their best were about 65% were caught in the early instars in the first trap that didn't make it up to the second trap. And then Muhammad, if you could mute yourself, please. Thank you. And you can see that across time, the sensitivity of the traps goes down across the way, especially as you get into the August period with the adults. They have the ability to hop right over them. But a trap is there 24/7 and doesn't need to be um, a trap is there 24/7 without having to have a person there to monitor. So even if they're not the most effective thing as a control device, they can be effective for finding new populations in new places. This was the slide I was looking for. Uh, at that nursery in Berks County, the first year, 12,000, second year, 7,600, 1,200, and the last year, it wouldn't even register. 2022 wouldn't register. Meanwhile, you saw on the sticky bands in the trapping site, population 6 miles away was up through the roof. Another thing that we see here, and this is kind of cool, there's something going on here that we haven't figured out yet. When we caught all these insects, the adults with the traps, we sexed them out. And you can see that the males would get to the site earlier and the females would follow. And we saw that the first year, 2019 with the long bars, 2020 with the dots. We had our traps up earlier that year because we had to install them the first year. We didn't have them out at the absolute beginning. And you see the sexes are even, and then we see this break. The males start heading out, and the females follow along later. And we saw that again, even though the population was much lower down here, we saw the males head out, the females follow along later. And to me, that tells me that there's got to be some way that they are signaling each other, even if it's not pheromones. From an evolutionary standpoint, it seems extremely unlikely that one sex would head out without the ability for the other sex to find them again later for mating and reproduction. Just a just a tidbit there. There's more there. We just haven't figured it out. And so we see the little commonality between relatively close locations, and unfortunately, we see that they keep hitting grapevines. Vineyards get absolutely slammed by these. This is Berks County vineyard that the population had decreased significantly in 23 and 22. Last year, they came screaming back in, and this is what it looks like when a female is laying her eggs. My son actually took this video. There's the female laying the egg in the vineyard. Just I thought it was a great video. And you can see on the end post, these are all individual egg masses. Like I said, they tend to cluster together, the females, put them in the same places. This picture on the left, you can see a clear size difference. The male is on the right, the female is on the left. The female is larger, and you can see what looks like these little black scars in the vine is probably feeding damage into the stems. And all this liquid, that's not rain. That is the honeydew from the insects. They're feeding such volume out of those vines that they deplete them. They reduce their ability to overwinter, and they are deadly if left unchecked in grapes. When we saw that in the summertime, we came back this winter and we did some preliminary sampling. 3 rows sampled in there. We found an average of 8.7 egg masses per vine, average of 10 egg masses per linear meter, excluding the end post because a lot of the end posts have been scraped. The eggs have been scraped off by the workers that were there pruning. But that worked out to about 1,594 eggs in 3 rows by 30 eggs per mass, 47,000 eggs in 3 short rows. Assuming a 70% hatch rate, which is not out of line, actually slightly on the low side sometimes, 33,474 nymphs can be expected from those 3 rows. In that vineyard, what that works out to be per acre, 508,000 per acre, 356,000 nymphs to hatch out at a 70% rate. 18.2 acres of vines in that vineyard, they can see 6.5 million nymphs hatching in 2 weeks.
Oh,
And
smokes.
we went back and we've actually since I've actually been working on the new numbers. What you're looking at is egg masses piled up in the vines, down in the trunk of the vine in that picture. The numbers held through the vineyard, unfortunately, and I think that might actually now be a low estimate. That's one of the things I've been trying to work on this week. So, despite overall populations in the core area where this bug started, we see that it's not gone and it remains a threat to grapes. And so, what we're tending to see now is that across the board, years one and two, years three and five, asking people where they are, what, knowing where people are in the infestation cycle, years one and two, and what are these things? They're so pretty, and that's where it's first starting. And then you get to that biblical plague. Is this a big biblical plague? We get asked this question with some frequency, and then the population drops off. It's not gone, but it drops off, and people say, "Where are all the lanternflies?" And then it seems like we're into this rinse and repeat cycle. And that's what I can say 10 years in from where it started. We saw it at the beginning and we have been watching this. Uh, Again, those vineyards are just hit consistently. This is what it can look like on an adult and during the adult phase. And these vines just cannot take that. Just some quick stats for Pennsylvania. We are the fourth largest wine production, fourth largest grape producer in the nation, and in combination with New York State, the Lake Erie grape industry, there is this unique microclimate for grapes along Lake Erie. And 32,000 acres of grapes in production between Western New York and Pennsylvania, largest grape growing region east of the Rockies, more than double the rest of the grapes in the entire eastern U.S. combined. And that is a huge production area for Concords for juice wine and table grape consumption. These grapes that are there are not grown the way wine grapes are grown. They are grown as a commodity, and they do not have the money to put into the sprays that the winery growers have that they can afford to put into the sprays. Everything, I shouldn't say everything. A lot of what Pennsylvania and New York is now geared for is trying to keep this out of that Lake Erie region and also to help them manage it when it gets there. New York wine and grape industry, 6.6 billion to the economics. Um, we're getting kind of toward the end of this talk here. I'll cut out couple of things. The other big thing, that's just Pennsylvania. Obviously, Pennsylvania and New York, when you think about wine, you think about California, grape growing. And so Dr. Melody Keena from the U.S. Forest Service, through with her growth chambers, climate, uh, ran ran her colonies at the climate of Stockton, California, in the Central Valley. Um, it not just the grapes, but 250 different crops, 17 billion per year in crop production. And the question is, are they going to be able to live there in that heat? Are they going to be able to handle that? Or I've heard people suggest it's going to be too hot and dry. And what Melody found is that no, it didn't matter. The week-long heat waves of 40 C max temperature did not impact the lanternfly survival. It may have impacted the egg hatch, the eggs that were exposed to that much heat, but it did not impact the lanternflies. There's nothing that has been seen yet that suggests that this is not going to be able to survive in California, and those growers are a little bit terrified, rightfully, of this getting there. So this is why we say do your best. Don't hitchhike. Don't let it hitchhike with you. Don't take it with you by accident if you're moving things. Um, we did see how it got to Indiana. It was a camper moved from Berks County, Pennsylvania. That was the first introduction. There were eggs on that camper, and it got established in Indiana. That's how it makes those big jumps. So, no evidence that disqualifies that possibility of it surviving. This is from Dr. Julie Urban, who is an evolutionary biologist that I work with who specializes in Fulgoridae. All other plant hoppers have five instars, not four. Spotted lanternfly is the only one that has only four instars as nymphs. And we also see that it has a very long adult period compared to the other plant hoppers. And we don't know why that is, but we know that they move around a lot. Uh, when they start getting bigger, that yellow abdomen starts to expand. And Julius found that that is a very good approximation of sexual maturity by comparing it with the dissection. And we also see that 40%, if you look at this graph, 40% of the samples were already mated despite not being completely expanded out in the abdomen. which means they are still capable of flying. They're still capable of moving, despite papers that were published suggesting that was not true. They absolutely are. Um, I'm gonna speed up through a couple of these slides here. The relationship there correlates very well, and you can see that as that abdomen gets bigger, the closer they are to laying their eggs, and they can lay multiple clutches. But way back in the beginning, when I was showing you the Atlantis cages, this is what the females that are successfully raised in captivity tend to look like. These are what the females that are wild caught look like. We still are struggling to keep them alive and happy and healthy in captivity that approximates what's going on in the wild. And so, with the necessity to go out and collect wild specimens to confirm what we think is going on, it keeps us pretty busy. There's a paper that was published. I was actually on this one too, based on what we were observing in Pennsylvania. Dr. Dennis Calvin put this together, and it shows where we think lanternflies would be able to successfully reproduce and exist successfully based on the number of accumulated degree days. Do they have enough time to get to sexual development based on when we see sexual development and egg deposition in Pennsylvania? And this looked pretty good. Uh, this is a map of Pennsylvania, the infestation in 2021. I like this one because you follow this line here where the darker red is where the municipalities where the lanternfly confirmed presence is, even if the entire county is in quarantine. And this happens to be the Norfolk Southern line. They've hitchhiked very well on the railroads. Railroads tend to have a lot of tree of heaven that grows along the tracks. But when we overlaid that with where the prediction was of where they were going to be able to reproduce it and succeed, it looked pretty good. It looked pretty good. Higher elevations, they weren't going to have enough growing degree days, and it would exclude a lot of places. And so Julie Urban went out, started collecting North Carolina, Lewistown, Pennsylvania, Nashville, Tennessee, and Rochester, New York. And she was comparing the sizes of the live masses recorded and 2024 in North Carolina and Tennessee, the adults much sooner than in Pennsylvania because they hatch much sooner. They get to adult. They tend to wait to lay their eggs till about the same time. Doesn't really matter so much where they are. But the later counts, you can see it makes sense. Pennsylvania in blue and Rochester, New York, yeah, they're a bit bigger than Pennsylvania. Okay, maybe moderated by the lake, like Lake Ontario. And you can see August 28th, September 5th, North Carolina, Tennessee is just bigger insects and multiple clutches. But then when we get to mid-September and you take a look, Rochester went screaming right past the North Carolina and Tennessee sites. And what that means by accumulated degree days, there weren't nearly as many accumulated in New York as anywhere else, and uh, seems to really indicate that something's going on. That this insect, the sexual development, seems to be happening independent of accumulated degree days in um, with the adult females. And so there's more to come with this. Dr. Julie Urban has been doing this, and this is an eye opener. And what that means is, that hypothesis, warmer regions, heavy feeding and reproductive maturation is going to occur earlier than in the cooler regions. And that's some more supported, right? They got there sooner. They became adults sooner in North Carolina and Tennessee. But that second hypothesis that the cooler regions reproductive maturation is likely going to occur later could be less impact from feeding, not supported. It's not supported, and it's really a bit of a puzzler why that would be going on like that. And so there's more to come. Stay tuned. I'm getting down near the end, so bear with me. I know we're running out of time here.
That's
Plant.
okay. Go ahead. Go ahead.
Okay.
Don't worry.
All right. So that plant health, what does it matter? This is a branch from a maple tree. As I mentioned, they like to feed on them. We did see some very odd, we've seen some very odd reactions to lanternfly feeding. Don't have it worked out yet, and I don't want to be too speculative, but when you shave back the bark, you can see what looks to be indicated that there's feeding damage going on into that xylem, definitely in that outer layer of phloem. And through the use of electropentrography. Dr. Holly Shugart at Penn State, that I am lucky enough to work with, is a histologist. She's done a lot of this work. This was first developed in the 60s for aphid feeding behavior to figure out how those stylets in their mouth parts work through the cells. And she's done some beautiful, I mean, these are just beautiful stainings of the salivary sheaths of how they feed down through the cells and get to where they want to be. And the Hemiptera, they have two types of salivary saliva used during the probing, the hardening saliva that makes that sheath, and then they have the enzymes that they inject in while they're doing parts of that probing, and it dissolves the plant cell walls and allows them to feed. It fights off some of that phytochemistry from the plants, the defense chemicals, and it can initiate a really strong host plant response, and that can maybe be explaining some of the damage that we're seeing in the feeding. And just an idea of what she's doing, uh, she's got these little bugs hooked up with tiny wires and gold thread wires that are measuring their feeding patterns and analyzing where they're feeding in the plant when different things are going on. And then she's working on the killing, staining, but these tiny threads are attached to the insects. And fascinating work, slow work, painfully slow work. More to come on that. Dr. Shugart is in the midst of that work. And so that's some of the new stuff that's going on that I think is really fascinating. Hopefully that will help us work out what's going on with that damage that we're seeing, why we see it in some species, not so much in others. And I will say this, the biggest question that I get asked is in the areas where the populations have dropped off, why is that? And um, we're not entirely sure, but I will say to watch for predators. So to a room full of entomologists, you notice the nice hexagon right there in the middle of all those lanternfly eggs, and we see that over and over again. We see the lanternfly eggs at the bottom, and we see our assassin bugs hatching out. And this is consistent from the time that they hatch. Those assassin bug nymphs are all over those eggs. They will feed on them right through adulthoods. Here you see that proboscis right into that thorax of the lanternfly, liquefying and having a juice box for a snack. There are a number of natural predators that I think have picked up on the populations have picked up, but unfortunately, we weren't looking at the populations before lanternflies got here. Again, uh, consistently, we see lanternfly eggs and wheelbug eggs and the general predators, the spiders and the praying mantises and whatnot. If you want more resources, our extension website has a fantastic amount of resources available. Um, to deal with the public. This is that outreach dissemination that you mentioned at the beginning that I'm part of. How to do these things, how to do it safely, to deal with businesses. Do they need a permit? Trainings. Tree of Heaven. Tree of Heaven is one of those plants you should not attempt to do without herbicide. You should not attempt to remove without herbicide. Uh, Management in vineyards. Work with the viticultures and uh, the management guide. I'm partial to that one because I'm a co-author, but I think it's a really solid bit of information in there. And my all-time favorite headline was I let spotted lanternfly survive, and then 50 tried to kill me. This bug has brought out the public like nothing I've ever seen before. These are acknowledgments for Julian Holly's work and for my stuff. Just so many people to thank. The top row, these technicians, um, they do just the best work, and they do it in areas where they are getting dripped honeydew on nonstop. And usually nasty roadside brush and full of ticks and broken bottles, and we couldn't do it without them. So with that, I will say thank you very much and stop my share.
Well, all right. Thank you. And everybody can throw up a reaction, hand clap. Yay. Very, very nice talk. Very informative, Brian. And at this point, I want to open the floor up for questions. Anybody have a question? Uh, please raise your hand and speak up. You can unmute yourself and talk if you have a question to ask. Okay. Or you can put it in the chat box also, either way. Um, there's a tremendous amount of information that we heard tonight. And I know there's some questions out there. I have a couple questions, but I'll let other people go first. So anybody want to ask, go for it. All right, then I'll ask the first question.
Okay.
So you mentioned um, injecting the trees with dinofuran, I think it was, right?
Dinotephuron. Yep. Teffuron. Yep.
Dinotep. How do
Yep.
you inject the tree? I was curious about that method. How is that
Sure.
done?
So there actually Dinotephuron can be applied a couple ways. It's incredibly um, soluble. And so you can actually apply it as a soil drench at the base. You can spray it on the trunk, and it's translaminar. It will move right through the bark and then get absorbed up. Or uh, if it, and the injection is more for tree care professionals. We actually have tools where you drill a hole in and inject it like an IV in a person.
Oh,
And so you can use a very small dose. The nice thing about that is it's completely contained in the tree, and the only way anything can access it is by feeding on the tree. So we avoid killing beneficials. Obviously, you can't use it before a tree flowers. There's restrictions to it, but because of the injection being so contained in that tree, it allows us to ignore the amount of active ingredient that is allowed per acre per year for treatment. Whereas if the other application methods, you are restricted how many pounds of active ingredient per acre per year. With injection, you don't have to worry about it. It's in the tree, it's locked in, and that's where it'll be metabolized. So, yeah.
very cool. And then for other applications like
Mhm.
grapes and I guess the grapes would be the number one. What kind of insecticides are they using? What is their choice?
It depends. So with the grape growers, they have to be very mindful of their harvest point,
Yeah.
and their insecticides are going to have called a PHI, the pre-harvest interval. And so depending on how close to harvest is going to determine what they're allowed to use. Mostly they are relying at this point on synthetic pyrethroids. So your bifenthrins, zeta-cypermethrin, and depending on the formulation, those those products, fenfopraxin, I think is a 30-day PHI. Bifenthrin, you're getting down to 20. Zeta-cypermethrin, some of them are 1 day PHI. But
Okay.
the
Yeah.
shorter the PHI, the longer residue, obviously, residual action. So some
Mhm.
of them are almost more like just a knockdown pre-harvest. And so might have to make that application, every several days, just to keep the populations under control. Once they harvest, then they can use the big guns again and with the longer residuals. But this is something new for growers to be putting out insecticides like this after they harvest. This is additional input on the crop that they've never had to do before.
Yeah.
And
And
that's an
expense, right?
expense. Yeah.
Yeah, absolutely.
Yeah.
So the pyrethrins, would I, if I understood correctly about them, they're more innocuous type of overall compared to other pesticides. They're a little less problematic, would you say, for people to,
Py py
uh,
Pyrethrins, yes. Uh, the synthetic pyrethroids can be problematic.
Okay.
The synthetic pyrethroids, all of them, they're non-selective. So you're going to take out your good guys too, your
Hmm.
beneficials. And so the more you're using these things, you're going to take out your beneficials that might help keep the mites under control, or you can get these explosions, unintended explosions of other problems because you've taken out the natural controls that would have kept them in line. So uh, I wouldn't want to say innocuous. Um, they're not as yes, they are the replacement for the organophosphates,
Yeah. Yeah.
right?
Mm.
But organophosphates are still in play. Malathion, um, they're still in play.
Mm.
So it's
Mm.
a it's more complicated than that is the answer.
I guess what I meant by innocuous, more for consumption for people in that regard.
Probably.
Yeah, the fight. Yeah.
Probably. Not my area of expertise. I go by what the EPA's telling us and what the registration is telling us. Um, I, I do I do think that we'll just leave it at that. Not my area of expertise. I have opinions. They're not important in this conversation.
Okay. No worries.
Yeah.
That was an aside.
Yeah, no, it's a and well, Dinotefuran, back to the Dinotefuran, it is a neonic, right? And neonics have that horrible bad name right now that they're killing all the pollinators. And we've done some work with maple flowers the following spring. What's the residue there? And it's sublethal. It's sublethal to even the most sensitive solitary bees. It's there's not enough residue to be on the lethal dose curve. Sublethal effects, remains to be seen. Unfortunately, we haven't been able to secure the funding to continue that out further, and that's been a frustration. But the initial work that we did collecting flower samples the following spring, if it's done after flowering and there's enough time for metabolization of that plant, it looks like it's not a problem for the pollinators the following spring.
Mhm.
I'd love to see more work done. Initially, I will say the work we did suggests it's sublethal. Now, there's more to be done.
That's good to hear. Yeah.
Yeah.
Who else? Some more questions? Well, we have a couple things in the chat. I'll mention that.
The
Um,
slide, yep.
and one person's asking if you would give this talk to the Carroll County Master Gardeners.
That's possible. If you get a hold of me and we'll see what the schedule looks like. We're getting into the busy season here with Hatch, but
Mm.
it's possible.
Okay.
Um, yep. And I'll put my email into the chat down there.
Hmm. So another question was, I have two other questions for you from the chat. Uh, Bill Murphy asked if you could put the slide on that shows the distribution in Indiana.
I can do. I don't have just Indiana. I have the big Cornell one that shows the whole Northeast. There's quite a number of counties now that are included in there.
Yeah, there was that one that showed so many more sites than the last one I saw.
Yep.
Both one site was on the Ohio River, um, probably lanternflies on detritus or something coming downstream, and the other was Fort Wayne, which is a big trucking hub that I was just curious to see. Yeah, so
Yep.
you can get this from Cornell.
Yeah, Cornell IPM maintains that. So if you just Google Cornell IPM spotted lanternfly map, they maintain this. Um, And like I said, I know Fulton County, Georgia is now included, and it's not there yet. And there's been a number of finds of individuals or eggs intercepted on the West Coast that there's no, this is showing where there are breeding populations established. So, yeah, no, I did a talk out in Purdue in January. Um, Lovely time to be in Indiana in January. And the there's the initial find there, I believe, down in this corner was the camper. I might be wrong. It might have been the one on the river. One of the ones in the southeast corner. It was a camper moved from Pennsylvania with eggs on the camper, and it got out there and they hatched. So the other finds there, trucking, shipping, trains, who knows? But that's how it's been making the big jumps. So I saw here, Julie mentioned the talk several years ago. Someone was fortunate, yeah, such natural enemies. So that's ongoing. Hannah Broadley at USDA has been still working on that. There are several uh, predatory wasps um, that will attack lanternflies fairly specifically. I think it's Dryanus sinicus was the one that looks like it's a strong potential. Um, don't hold me to that. One of them that attacks the nymphs has a strong potential uh, for possibility of release. It lays its egg in the nymph. The egg develops in the nymph, and then it breaks out. Pretty wild science fiction looking stuff. I believe the egg parasitoid was not specific enough. Again, don't hold me to this. Um, but I'm pretty sure the nymphal one still looked pretty solid. Also, Dr. Mari West from UC Davis has been involved with a group. I met her. I know there's other people in that group. They've gone and collected lanternflies, not spotted lanternflies, but lanternflies in the Southwest that actually identified several new species of predatory. Um, I think wasps that by collecting the eggs, putting them out, then um, they were able to collect the predatory insects that were attacking them, and they've since taken the ones that were attacking the native lanternflies, hatched them out, and one they had all males, couldn't reproduce with that. The other one they had males and females, and then put them on spotted lanternfly eggs, and they seem to be doing quite well. And because they're already native in the United States, there's a whole lot less regulation to worry about with that one. So that's another avenue that's being pursued. That's the UC Davis folks working on that. And mm, unfortunately, I don't have better details off the top of my head, but it's ongoing. It's ongoing. Yeah.
I'm not sure who's there at the Entomological Society of Washington, but I'm glad somebody joined from there. Um, Perhaps they could say who it is. Gary Hevel, perhaps.
It's
I
Alan.
don't. Huh?
It's Alan Norbaum.
Yeah,
Oh,
that.
hi, Alan.
Hey,
Hey,
Al.
Alan.
Hey,
Hey,
Alan,
Alan.
Cool.
everybody's best friend.
Thanks for joining us. Yeah.
Yeah, no, it's a good talk.
Did
Enjoying.
I answer your question good enough about that?
Yeah, I mean, looks like there's a lot of things
Parasitoids?
going on in general with
Yeah.
that, which you expect, but you guys are.
It feels like we're running backwards sometimes, but we've come a long way and not far enough in 10 years.
Mhm.
Yeah. So
So it's 8:20 and Al is still at work. Those government employees just fraud, waste, and abuse, I'll tell you.
Well,
Yeah, really.
I stayed later to listen to the talk, so
The
Wow.
true dedicated entomologist.
mm. Hey, I'm just a landscaper that they fed me and I kept coming back. So what can I say?
Brian, if I may,
Yep.
you said, "Oh, I only have my AA."
Hmm.
I have to tell you, it just occurred to me, it's not degrees that make a great scientist, it's curiosity.
Hmm. It also
And
killed the cat, though,
your
you
talk
know?
was your talk was fantastic.
Thank you.
Yes,
Thank you.
indeed, very good, Alan. Thank you for saying so because I felt the same way too. Uh, but you put it into succinct words that very true. So yeah, Brian, fabulous talk, and you
Thank you.
covered so many things, uh, and I didn't expect so many facets in the talk. So thank you for all that.
You
Um,
gave me free reign, so I ran with it. So,
hey,
yep.
absolutely, that's what we like. Yeah. So anybody else want to ask anything or say anything, please feel free. Um, and then we can let Brian off the hook here. So nobody has something more. I mean, because he covered a lot of stuff, and I was uh, I was going to ask about any other natural controls, but you just mentioned all of those, so that was good. Uh,
There's the one big one that is Bavaria bassiana, and that was the fungus that we found in 2018 that was killing them. Well, we find it every year late in the season. Uh, there are commercial strains of it available. We actually applied it by helicopter in National Rec area, and Dr. Nina Jenkins, formerly with Penn State, has developed a product that uses that for control of bed bugs indoors. We use the indoor product. We use
well,
the biospheres. There's a number of commercial formulations available of the Bavaria bassiana, and we have not been able to get consistent infection with it. It's not happening. We can get as good as 46, 47% infection and as low as 2%. And the ones where it was 2%, we thought we had the perfect weather conditions. Uh, for whatever reason, we can do it. You spray it on directly. It's great. You can get pretty consistent results. In the outdoor trials, we just have not been able to get consistent in infection with it. And so for that reason, we don't recommend it for growers, for government agencies to go and control until it can get tweaked out and get more consistent results. Dollars and cents, it's not worth it. So that's the other big native one that we usually that we've looked at. Um,
what
so
did you say the name of the fungus was again?
Bavaria
Bef-
bassiana. Yeah.
Okay, yeah, I think I heard of that.
Yeah. There was another one that was identified at the same time in 2018. Now, 2018 in eastern Pennsylvania, we had double our annual rainfall. It just rained nonstop. And this other fungus, it was found by a Berks County Parks uh, employee at Antietam Park, just south of Reading. Betcoa major was identified as. And that one was funky. It was like a zombie fungus like reaching out from the trees and attaching to the feeding insects. And unfortunately, we've not seen it since it's not been that wet again. I'm sure it's out there. It's just not as prevalent, and there's no commercial formulation on that one. The
Hmm.
Bavaria bassiana, there's quite a number of formulations on it.
So
So
is Bavaria a more broad spectrum type of thing?
it is. It is, and there's also some evidence that the lanternfly are um, able to self-regulate their body temperatures and ward off the infection. And when we generally see them succumb to it in the wild, and we'll find the wild. It's a native fungus in our region. When we generally find that they've succumbed to it, it's post egg laying. They're depleted, they're worn out. That's when we tend to find them infected. And it looks like, given if they're healthy and they're doing well, there's some indication that they're able to ward it off. Um, so yeah, unless they're absolutely saturated in it. Yeah.
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 All right. Well, Brian, in appreciation for your talk with us tonight, we want to extend to you yearly membership to MES, so you receive our journal and our
Nope.
newsletters,
Thank you.
right? Um, and we want to send you that. Um, so if you could, perhaps I could get your preferred mailing address so we can send that along to you, right?
Sure.
And we also, I don't know if I mentioned this, I talked about it with Sarah, your colleague, about an honorarium, but she said that something about that's,
Not necessary.
well, we want to offer that to you as well, you
No, I appreciate it, but I traveled all the way to my living room, so.
Hmm. Where are
But
you?
I'm in Berks County, Pennsylvania. So about half an hour, about 30 miles north of Philadelphia, and I'm stationed in Berks County for in the extension office there. So
So you're near Allentown somewhere?
yeah, just west of Allentown. So Allentown, Reading, Philadelphia, we're kind of dotted in the middle. Our Berks County office is right outside of Reading.
You know where Hawk Mountain is, right?
Yep, there's a vineyard. That vineyard where I was showing all the egg masses. I can see Hawk Mountain from there.
Would you
Oh,
consider that? You were talking about how the spotted lanternfly did poorly at higher elevations. Is that
it,
a higher elevation, the top of that ridge, Kittatinny? No.
no, we're talking up, um, quite a bit higher, like the Allegheny Forest in central Pennsylvania, Scranton, Wilkes-Barre, that into the Catskills, Adirondacks, definitely that kind of elevation difference. Yeah. Yeah, the northern plateau of Pennsylvania, which is Bradford, Sullivan counties. Bradford's the ice box of Pennsylvania. It looked like it was going to be too cold. Well, I think it just got added into the quarantine 2 weeks ago. So, uh, yeah, and there's always that big question here. Just because the just because the average temperatures suggest it shouldn't. We've been so warm the last couple of years that it doesn't take. There's not a lot of swing from the far north of Pennsylvania to the bottom end of Pennsylvania. It doesn't take much of a change in accumulated degrees to see that. Am I making sense? To see the southern temperature average in the north. It's not that big a swing, if that makes sense. Um, yeah, we're looking at averages. Will those populations maybe be knocked back some if we get no more average year? Probably. But Julie's work with the Rochester site really indicates that there's something else going on beyond accumulated degree days. And that's pretty interesting. Um, what signaling it? Is it the change in the trees, the senescing of the trees? Is it that the trees are moving that much more nutrients available in the phloem? What's going on? But why is that those North Carolina populations, they get to adulthood and then they just sit and wait and wait and wait and wait to lay those eggs. Um, Crazy. It's a stinker of a bug.
know.
Yeah. Hell of a first invasive to pick to work on my end.
Yeah. All right. Well,
There,
One
there-
crazy question.
oh, there
Brian,
we go.
you know Amy Corman?
oh, I do. Love her. Talked to her this morning on the phone. Yes, and Amy is the one that gives me reassurance that says, is if you know Amy, she's been around the world and seen them all, and or many, and she says this is the damnedest bug she's ever seen. So that makes me feel better.
When she was a junior captain in the Army, she worked for me.
Is that right? I'm writing your name down. Anything you would like to pass along?
Just tell her I said hello. I saw her at the Eastern Branch meeting up in Harrisburg last month.
Very
So,
good. Very good.
she is a great person.
And she is a sounding board when I'm stuck in my thought process. She is one of my sounding boards who starts asking questions and shooting it back to me. So yes, yeah, she's fantastic. Cool. All
Very
right. Well,
nice.
thank you much for having me. I'm gonna go grab some dinner with my family.
Brian, thank you so much for speaking for us again, and we'll
You-
be in touch, okay?
Well, very good.
Take,
Thanks.
take care. Thank you.